is right here. This is the starfish. I'll label it as starfish. This is the frog. So this is zygote, right? Fertilized egg, zygote. And then 2 cells is missing. Somebody took it and hang it up from bottom of the truck. You've seen those things that hang down? I guess that's a norm. But there was one. There was one that had 2. And then 4, 8, 16. So I will never ask you, what is this 4 cells, 8 cells, 16 cells. But if I show you this or this, these are morula.
Morula.
Okay, morula. And then after that, it is still, who knows, this is morula or blastula. I don't know, right? You don't know. So, but when you open it up, inside of it, right, they cut it for you in half. This is blastocele, right? So this is blastula. And I don't see any invagination. You know what I'm talking about. Now I see invagination. You remember that term I was using? The cells are still divided. Look, the cells are smaller than these cells. And they call blastomere. You know, the cells is called blastomere. All of these cells. So invagination occur here. You still have blastocele. This opening, this invagination right here. If I have a piece of tape here, that is called blastopore. You're familiar with all of that. The stage is gastrula. Gastrula, right? Am I making some sense? And then of course this is gastrula. That invagination is becoming more and more and more, right? This is later than this one. This is earlier stage. Okay, so it's becoming more and more. Now this is endoderm, of course. What germ layer? Endoderm. Blastocele, still. Blastopore, still. Same as this one, right? You know what I'm talking about. And there is one more question I can ask here. What is the name of structure? That is archentron. Okay, this is archentron. That's it. That is all of the starfish. And I don't have Bipanaria larva in here. You guys have it in your slides, right? Frog, zygote, right? It has a thick yolk on the bottom. The white is yolk. And then 2 cells. Nobody took this to hang it from the truck. Okay, but anyhow. And then you have morula, a few more cells, morula. And then this one is blastula. Look at that opening right here. So this gotta be blastula. Still, this is blastula, if you would, right here, because of blastocele, 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 blastula, and then still blastocele, C-O-E-L, it means space, right? And that is a space full of fluid. You know, remember that? This is not empty space, it's a fluid field. Okay, so, and then the next one is gastrula. So, because you have 3 germ layers now, the red, orange, pink here is mesoderm, endoderm, ectoderm, right? You all know, so this is gastrula. What stage gastrula? What germ layer? Then you should know that. This could be archentron. This is archentron. And then same thing, it's a yolk plug. The name of structure right here is yolk plug. You do have them on the other model too. It's in here somewhere, yolk plug. But anyhow, so this one, they cut it longitudinally. So this is notochord, the pink. And then this is archentron, endoderm. The orange here is mesoderm, yolk plug, endoderm, ectoderm. Same thing. Now, this is neurula stage. They're different than this one. Neurula, and this structure right here is called neuroplate. Okay, so then they cut it in half for you guys to see inside of neurula stage. This is neuroplate. This is neurula. So you have mesoderm. The pink is notochord. Okay, do you see that? The pink is notochord. The orange is mesoderm. Yellow is endoderm, and this is ectoderm. Okay, those are germ layers. The question for that would be germ layer. Then this is gastrula. Remember I said gastrula and neurula, and then now this is very early embryonic stage. Okay, embryonic stage. And what you have here is neural tube. The blue is neural tube. The pink is notochord. Then underneath this would be archenteron, the embryonic gut, right here. The blue is inside of it. So, and the yellow is endoderm. The blue on top is ectoderm. The pink is mesoderm, and on the very bottom you have mesoderm here too. Okay, so this is the actual tadpole now. You all know what tadpole is, embryonic frog. Same thing, neural tube, ectoderm. Pink is notochord, archenteron, everything is in there. And right here, they cut the tadpole in a cross section. Okay, so these are the eyes of the tadpole. They cut cross section. So you have neural tube on top, notochord on the bottom. You have somites now, mesoderm, endoderm. The yellow is endoderm, and that's pretty much it. This model, these three, is also your frog. So this is gastrula, right? And then in the gastrula, the green is notochord. Then the yellow is endoderm. The blue is mesoderm. Then this is the embryo stage, early tadpole. Same thing as here. The green is notochord. The blue is neural tube, endoderm, right? And then the orange is mesoderm. Same as this one. Same thing, but the notochord is green. Mesoderm, neural tube came from ectoderm. What germ layer is ectoderm? What is the name of structure? Neural tube. Do it make sense, everybody? The archenteron, endoderm, mesoderm, and that's it. Okay, check. This is 33 hours check. This is the whole mount, the whole entire check. Optic vesicles, prosencephalon, mesencephalon, rhombencephalon. Somites, all of these are somites. You guys see that? Those are somites. Neural fold and neural groove. Inside is neural groove. Then you have the foregut, the dorsal aorta here on the ventral side, heart, dorsal aorta, somites. Oh, the orange line, you see that on top of the blue? That is notochord. So the blue underneath is neural tube. It's a tube, okay? And this tube at the end, the neural tube at the end, at the posterior end of the animal, it opens up, becomes like this. So they have neural fold, neural groove. Do I make sense, everybody? On top, it's a tube. Does that make sense? I hope that is clear. Okay, so the serial section, they cut the animal through the head, they cut the animal through the heart, they cut the animal through somites, and they cut the animal through the posterior end, right? So they cut, am I making some sense? It's called serial section. So through the head, optic vesicles, prosencephalon. And then of course, the red, orange is mesoderm. Then pharynx, dorsal aorta, neural tube, underneath is notochord. This is through the heart. So the outer layer of the heart is epimyocardium. The inside is endocardium. I might ask you what is specifically, what is the specific name of structure? I might ask you what is the name of the organ is heart. What is the specific name? Epimyocardium. What is the name of structure? Be specific, endocardium. Pharynx, the yellow, dorsal aorta, neural tube, underneath notochord, coelom, the black area is coelom. Okay, and these are all blood vessels right here. So those came from mesoderm. Same thing, heart. And then right here, the posterior end of the animals. Then you can see the somites. This is a somite, neural tube, dorsal aorta, and then you go farther down, then you don't see the tube, you see the neural fold, neural groove, and somite again, notochord, that's it. 33 hours. Amoeba, This would be a pseudopod, the whole entire thing. I put the tape all around it, that would be pseudopod, 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 pseudopod. And then what you have the cell membrane, the blue is ectoplasm, the yellow is endoplasm. Do not confuse it with ectoderm, endoderm, two different words. Cytoplasm, phagocytosis occurring here, food vacuoles, nucleus, contractile vacuole. And that's all I have here. On this one, same thing again. You have pseudopod. The very outer layer is ectoplasm. Those gray area is endoplasm, cytoplasm, contractile vacuole, nucleus, lysosome, food they ingested. These are bacteria they ingested. Paramecium, phylum Ciliophora, phylum Amoebozoa. You have to know the name of the phylums. Macronucleus, micronucleus, oral groove, cytopharynx, cytostome, ampulla, contractile vacuole, cytopage or cytoproct right here, ampulla, and contractile vacuole. This one, same thing, oral groove, pharynx, cytostome, ampulla, contractile vacuole, micronucleus, food vacuoles, cytopage or cytoproct. See, they're emptying out the solid portion to the outside of the animal. Ampulla, contractile vacuole, cytoplasm, cilia. Though of course these are trichocytes which hold the cilia in place. They did not draw the cilia. So the cilia is outside, but these yellow ones are trichocytes. Am I straight? Am I done? Okay. Have fun. Enjoy.